Yeah, welcome back. Now I hope you have you gained energy during the coffee break. And now Wendy is continuing his second talk following the first presentation.
Thank you. So in my previous presentation, I spoke mostly about genetic discovery, using genetics and genomics and bioinformatics to discover genes. And this is what I've been doing for the last 15 years or so in disease resistance of cereals. About 5 years ago, I moved from the UK, where I had my lab in the John Innes Centre. I moved to KAUST. And with the generous funding that I now have at my disposal in KAUST, I set up 2 additional pillars of research in my lab, which are depicted here. So we have our discovery and function. We still continue that. And then we're also venturing out into mechanism. This is a very new area for me. And we're also now starting to do some engineering. And then finally, we have a pathway to impact partner for our deregulation and deployment. And so in this second presentation, I will talk about some of the work that we've been doing in KAUST over the last few years in the area of mechanism. And so by way of background, I mentioned in my previous presentation that most of the genes for disease resistance that have been genetically defined and subsequently cloned, they belong to 2 classes of resistance gene structures. They typically encode surface receptors consisting of leucine-rich repeats that act like aerials on the surface of the cell, monitoring the environment for the presence of non-self. These leucine-rich repeats are protein-protein interaction domains and can detect the presence of pathogens based on effectors that they secrete. And then they have a transmembrane domain, and some of them may be decorated with a signaling domain, a kinase. And then you have a second type of receptor, which is an intracellular immune receptor, a so-called nucleotide binding site leucine-rich repeat domain. And if we look at the number of genes that have been cloned over the last 30 years since the first one was cloned, you can see this is a sort of slightly exponential growth. And to date, more than 400 disease resistance genes have been cloned. And we looked at all of these genes and quantified the structures that they encode. And as you can see, most of them are cell surface receptors or these intracellular NLR receptors. And then you have some oddballs, some other things. And we've seen that there's a class which is emerging, which are these kinase fusion proteins. And if we look at where these disease resistance genes have been cloned from, it's interesting to note that while many genes have been cloned in the model species Arabidopsis, the cereals are now leading the way. And these are primarily genes that have been cloned in wheat and corn or maize. And I find that very gratifying now that we can do a lot of this work that was only possible before in Arabidopsis. We can now branch out into the major crops and clone these genes. And here's another analysis that we submitted when we wrote this review. We submitted it to the journal, but one of the reviewers did not like it. It set off a lot of antibodies. And this is where we quantified the impact of, the publication impact of cloning these genes. And so on the y-axis, you have the citations normalized by the number of years. And I'm just trying to say that there's still a lot of interest in cloning these genes, and you can be creative in the way that you publish them and still publish in good journals. Okay, so by way of a little further introduction, and this relates, I think, to the question that Staffan asked at the end of my first talk, and that is, how do these receptors work? And the most simple model is that they detect the effector from the pathogen. So the pathogens, they secrete a lot of proteinaceous molecules either into the apoplast of the leaf, or they have ways of injecting these proteins into the plant cell, and they promote pathogenesis. They subvert the cell and help the pathogen to grow and reproduce. And of course, the plant has evolved these immune receptors that can detect the presence of the pathogen and elicit a defense response. And sometimes this is a direct interaction where the immune receptor will recognize the effector and elicit a defense response. But a very important contribution to understanding what's going on was made by Eric van der Biesen, a postdoc in Jonathan Jones's lab in 1998, and he proposed the so-called guard hypothesis. And in this model, the effector is not recognized directly. The effector is interacting with a pathogenicity target, which is shown in orange, and the immune receptor is guarding this pathogenicity target and can detect modification on the pathogenicity target imposed by the effector. And this can then turn on downstream defense responses. And this is a very nice model because it explains how millions of effectors from lots of different pathogens can be guarded by a limited number of immune receptors. A typical plant genome will have from a few hundred to a few thousand immune receptors. And if you now picture in your mind that these immune receptors are guarding cellular hubs, there's only a finite number of hubs that the host would need to guard. And a corollary of the guard hypothesis is the so-called decoy model. And in this model, the pathogenicity target has been duplicated in the genome, and that releases it from the functional constraints that it may have. And so now the immune receptor is recognizing a decoy, and the decoy is acting as a trap for the effector. And finally, in the integrated decoy, the decoy is physically integrated into the gene of the immune receptor. So you have the decoy and the immune receptor right next to each other, physically next to each other. And that occurs in about 10% of NLR. So 10% of NLR immune receptors, they have integrated decoys. And this tells us a lot about the types of pathogenicity targets that effectors they target. Okay, so we started off years ago cloning disease resistance genes in wheat. And I will admit that at the time I was expecting that we would clone NLRs, these intracellular immune receptors, which are the most abundant genes that have been defined by genetics and cloned across many different systems, including in monocots and dicots. And to our great surprise, many of the genes that we cloned in cereals, they did not fall into this class. Instead, they were kinases. Very, very often they were tandem kinases, so two kinases fused together. And sometimes they had an altogether completely different exotic domain fused to them. And we call these class of genes now, we call them kinase fusion proteins. And of course, we were wondering, how do they work? Right? What's the mechanism? What's the modus operandi? And so that is what I will talk about in this presentation. We have focused our attention on this gene here, SR62, and I'll take you through how we studied SR62 and determined its mechanism. So SR62 is a gene that was introduced into wheat from a wild grass, Aegilops sharonensis. It confers very broad spectrum resistance to stem rust, so it's a very good gene. And it was cloned by a postdoc in my lab, Gutai Yu, and we published that in 2022. And as part of the, this looks a little bit corrupt, this slide. Anyway, as part of the gene cloning procedure, we generated EMS mutants. And you can see the effect of these EMS mutants on the mature protein up there. And SR62 encodes a wheat tandem kinase. But as part of cloning SR62 and generating these mutants, we also found a few mutants. We found 4 mutants that did not have a mutation in SR62. And these are depicted here, 119D, 200H, 267D, and 1298E. The other 2 are controls. We've got the resistant parent, so here's 1644, which carries the intact gene, and on the far left. As you can see, that's completely immune. And then we have the recurrent parent, so here is completely susceptible. And the mutants, they are as susceptible as Sahir. And so the question was, if they don't have a mutation in SR62 in the kinase, what are they mutated in? And my former PhD supervisor, Jonathan Jones, he has followed somehow in my footsteps. He has become an adjunct professor in KAUST, and he comes out to visit KAUST every 2 weeks. Every year he comes for 2 weeks, typically in December. It's very nice. We catch up. We talk a lot of science. And I was telling him about this project. We were sitting outside under the palms, having a lovely lunch. And I told him about this. I told him about these extragenic suppressors. And he said, if it's an NLR, it's a Nature paper. So, of course, I was very motivated then to clone this gene. And so postdoc and 2 students in my lab, Renji, Karthik, and Oliver, they set about cloning this gene. And to cut a long story short, we found that it was indeed an NLR. So the tandem kinase and this NLR, they occur physically close together in the genome as a digenic module. So we have the kinase and the NLR very close to each other. And these 4 mutants, we could indeed show that they were all in this NLR, providing very good evidence that this is the gene that is required for the function of the NLR. Okay, in parallel with this, we worked with Peter Dodds and 2 postdocs of his, Megan and Sean, in the CSIO in Australia. And at the time, they had developed a very good system for cloning the corresponding effectors of resistance genes. And they applied their method to SR62. And lo and behold, they identified the corresponding effector. So they identified AVR SR62. And AVR SR62 belongs to a multi-gene family in the stem rust fungal genome. And you can see in this strain of the fungus, on chromosome 5A, we have 4 copies, and on chromosome 5B, we have 3 copies. And I've highlighted the ones in green that are able to elicit the hypersensitive response, that are functional versions of the effector. And they were able to validate these in a protoplast assay. So they could isolate protoplasts from wheat and deliver the corresponding effector with the wheat tandem kinase and elicit a hypersensitive cell death, which is measured by quenching of a reporter gene that gives fluorescence in this system. And also, interestingly, they showed that the tandem kinase is the primary immune receptor. They used yeast-2 hybrid to show an interaction, a physical interaction in vivo between the tandem kinase and the immune receptor, and the effector. Notice that only the green flavors of the effectors, the ones that are able to elicit a cell death response, only those physically interact in the yeast-2 hybrid system. Okay, the next step was to functionally validate the NLR and its requirement in this system. And we used the protoplast system, and we were able to take mutant backgrounds that had a mutation in the NLR and deliver to these mutant backgrounds the NLR and the effector. And only when we delivered the functional NLR and a functional effector were we able to elicit a cell death response and quench the reporter gene activity. So this was very nice, a very quick way to validate the function of the NLR. And we were also able to recapitulate the gene-for-gene interaction in a heterologous system by transiently expressing these components in a leaf of Nicotiana benthamiana. And so here, in each of these sectors that are indicated by the circles, we have expressed the NLR and the tandem kinase. And if we express these two together with a negative control, YFP, which you can see at about 1 o'clock in the top right-hand corner, you see that there's no cell death. So just expressing the NLR and the tandem kinase, you don't get cell death. But when you co-express the green flavors of the effectors, you get cell death, but only the functional effectors, indicating that we can recapitulate the gene-for-gene interaction in this transient system, which is very useful because this is a very fast assay. Okay, so now I'm going to take you through 3 slides, which will give you an idea of how we dissected the mechanism. Just going to show you 3 slides, okay? So keep your attention for 3 slides. I'm not going to take you through the 70 pages of supplementary data that was in the paper, okay? Just 3 slides. So in this first slide, and this was an experiment done by Luby and Renji, 2 postdocs in the lab, they showed that in the resting state, the tandem kinase and the NLI immune receptor, they do not interact. So if we fuse the partners with a reporter gene luciferase, we do not get illuminescence when we express these, which is shown on the left-hand side of this leaf. Okay, so the kinase with the NLR does not interact in the resting state. But when we co-express a functional AVR effector, AVR SR62, then the NLR is recruited to the complex, and you get functional luciferase expression and light. Okay, so this is very nice. And again, it was a very fast assay to do. And when we then did yeast-2 hybrid and separated kinase 1 from kinase 2 in the wheat tandem kinase, we could show that it is kinase 1 which interacts with the effector. It's not kinase 2. Kinase 2 does not interact with the effector. Okay, so then we went on, and this is work done by Sean in Peter Dodd's group, and he showed that AVR SR62 is able to disrupt the interaction between kinase 1 and kinase 2. So first of all, he in this assay, it's a plant 2 hybrid assay in which you get the accumulation of a red product called betalin when 2 proteins are physically interacting. And when he expresses kinase 1 and kinase 2 separately in trans, he gets interaction, which is shown in the top right-hand corner. You get the production of this red pigment showing interaction. However, if you co-deliver a functional effector, then kinase 1 and kinase 2 dissociate from each other and you break that interaction. Okay, so to recapitulate, in the resting state, kinase 1 and kinase 2 are interacting, and that interaction is lost when you add a functional effector. But the interaction is not lost when you add a non-functional effector, as shown at 7 o'clock in the leaf. Okay. The other thing which is very interesting is that you can express kinase 1 and kinase 2 in trans along with the NLR immune receptor. And when you express kinase 2 with the NLR, which is shown at 11 o'clock, you get an HR. Okay, so kinase 2 is enough to activate the NLR and give rise to a hypersensitive response. However, if you co-express kinase 1 and kinase 2 in trans together with the NLR, that HR, it doesn't happen. So in other words, kinase 1 is negatively regulating kinase 2. And you can de-repress that regulation by delivering in trans a functional version of the effector, which is shown at 7 o'clock. When you deliver a non-functional version of the effector, that de-repression does not take place, as shown at 4 o'clock. Okay, so I said I would cut the mechanism short, and I'm going to go directly to the model that we came up with. And there were a myriad more experiments that we did, and the reviewers doubled the amount of experiments that we did. So this model is very well tested. And of course, you can go to the paper and you can read all the detail if you wish. But to cut this long story short, we found that in the resting state, the wheat tandem kinase exists as a homodimer. And in this homodimer, kinase 1 is interacting with itself. So it's interacting with kinase 1. And kinase 2 is interacting with kinase 2. And also kinase 1 and kinase 2 interact with each other. Okay, so they're tightly bound together like this. And then when you add the effector, it comes along and it interacts with kinase 1. And this releases kinase 2 from this complex. It's still tethered, but it's now floating about. And this exposes an N-terminal lobe in kinase 2 that allows it to recruit the NLR to the complex. And this recruitment happens through an N-bark domain, which is highlighted in red in the figure. And this then activates the NLR and leads to immune responses. And we coordinated our work with a group in China, in Beijing, and they cloned the same NLR as us. And so we were a little sort of worried about the competition, but actually it was a really, really good coordination, and we managed to publish back to back. And what was really nice was that they worked on the latter immune responses, and they showed that, so they worked on all the late molecular interactions, and we worked on all the very early molecular interactions. So it was very nice to have these two papers back to back in the same issue. And notice that this kinase 1, it has these long bits, and these are called extended beta fingers. So these are it's an extension of the beta finger in the kinase domain, and this extended beta finger is responsible for maintaining the homodimer as a complex, and it's also the bit that interacts with the effector. And so here's a result from our study that I'm very proud of. I encouraged Oliver, who's a bioinformatician, to download serial genomes, and he downloaded 70 serial genomes, and he managed to find 30 or so orthologs of the SR62 kinase. And he aligned all these orthologs with each other and was able to identify regions of the protein that are subject to natural variation. And when you project that natural variation onto the AlphaFold generated 3D structure of the wheat tandem kinase, which is shown here as a homodimer, what is really interesting to see is that the hotspot of variation occurs where the effector is binding. So it would appear that there's been selection for variation at the recognition surface. So there's really an arms race going on between the effector and the immune receptor. And if we draw the extended beta finger as a ribbon instead of a blob, and that's shown on the far right, you can see the beta 1 strand going out and the beta 2 strand coming back. And you can see that the beta 1 strand, which is the one that's buried in the complex, is rather conserved. And this is the one that is mediating the interaction between the kinases. Whereas the beta 2 strand, which is on top and which is solvent exposed and is the one that's interacting with the effector, that is the one that is really hypervariable. Okay, so then this is what Jonathan, my former PhD supervisor, remember this is what he said when he came to visit in December 2023. Then he came back to visit again in December 2024. And I told him, Jonathan, we've cloned the gene. It's an NLR. And then he said, it's a science paper. So, you know, he's getting old a bit. He's now 70. He's just turned 71. So maybe this was a senior moment. But anyway, we followed his advice and we submitted it to Science. And I was very happy. It was a positive outcome. Okay, so you may ask, you know, why all this mechanism? What's the relevance for breeding, in particular GM breeding? And so to remind you, SR62 was integrated into wheat from Aegilops sharonensis. And if you have the 2 sharonensis versions of the SR62 kinase and the NLR, they work very well together and they give rise to immunity. But if you were to put SR62 into a stack and express that in the cultivar fielder, it does work. So the fielder NLR is able to work with the sharonensis version of the kinase, and you can get immunity. But we have shown that if we take another flavor of the NLR from another cultivar of wheat called Sahir, these two do not work well together and do not give rise to immunity. So it's very important to have the two right combinations of this digenic module together. And so what we've learned from this exercise is that if we're going to build a stack, it's probably a good idea to include the kinase and the NLR together in the stack. Okay, so continuing a little bit further with this interesting feature that we discovered, the extended beta finger, we looked at other kinases that have been cloned as major determinants of disease resistance gene in cereals in the last 20 years or so. And it's not just us who have cloned some of these genes. The community as a whole, in the last 5 years, there's been a lot of these genes that have been cloned. And we looked and we found that in nearly all of them, except one, there's this extended beta finger in the kinase. So we thought it must be an important feature. And so Oliver, he built an HMM model, which is able to recognize these features in a plant genome and annotate the plant genome. And as you can see, there's quite good conservation in the first few amino acids and in the last few amino acids, and a little bit of conservation in the middle. But apart from that, it's a very variable feature, this extended kinase. The length is very conserved, but many of the positions are very variable indeed. And within the wheat genome, we've annotated all of these now in Chinese Spring, and they are scattered throughout the genome, mostly found in the telomeres, where in the gene-rich region. And many of them occur quite close to an NLR. So it gives us a possible hint at which NLR these kinases might be working with. And this is something we can now, of course, empirically test. And this shows the type, the phylogeny on the right shows the type of kinase fusion proteins with the known ones shown on top. Okay. Now, what's interesting is that some of these fusions appear to have evolved independently. So here we have shown, we've highlighted in green the kinases that are fused to motile sperm proteins, MSPs. And you can see two very distinct clades. This phylogeny is based on the extended beta finger. And you can see two very distinct clades, suggesting that possibly MSPs were recruited independently at least 2 times during the evolution of wheat. And here's another example of two kinases that are fused together. And this fusion also possibly occurred multiple times. And also interestingly, many of these extended kinases, they occur in kinases that are singletons, that are not fused to another kinase or to an altogether exotic domain. And they may be the progenitors that through evolution recruited these exotic domains, which we believe probably act as decoys, following the decoy model that I presented early on. So they're a bit like integrated decoys, but not for NLRs, but for kinases. And another very important feature that we've discovered is that if you look at the expression of these kinase fusion proteins in a plant genome, the ones that have been defined as being functional are invariably in the top 25% most expressed kinase fusion proteins in the transcriptome if you just look at kinase fusion proteins. Okay, so this is a hallmark, a feature that could be used to speed up the cloning and identification of kinase fusion proteins. Because if you have an interval that you have genetically defined and you then project that genetic interval onto a physical genome, you can annotate the genes in that region and look at their expression to help identify the best candidates. And if we look at the genomes of monocots, we find that this feature, the extended beta finger, is restricted to the Poaceae. So suggesting that the feature evolved about 100 million years ago. And this may explain also why disease resistance genes encoding these kinase fusion proteins have only been identified in wheat and barley and their wild relatives so far. And this work hasn't been published yet in a peer-reviewed journal, but you can find this story explained in this bioRxiv paper. And so a few concluding remarks. The SR62 kinase and NLR, they function as a digenic module encoding a sensor and a helper, where the sensor is the kinase fusion protein and the helper is the NLR. And kinase fusion proteins, including SR62, they have these extended beta fingers, which are near ubiquitous. We've only found one example of a functional kinase fusion protein that doesn't have this extended beta finger. And this feature is private to the Poaceae, and so most likely evolved about 100 million years ago. And functional kinase fusion proteins have so far only been cloned from the Triticeae. So that's the group of grasses that include wheat, barley, and rye and triticale. And once again, this is the acknowledgements to my group who conducted this work. And I'd also like to thank again Peter Dodds and his group in the CSIRO in Australia, and the very good collaboration and coordination that we had with our friends in Beijing, Xiyong Lu and his research scientist Ping Lu. And thank you for your attention.
Yeah, thank you, Brande, for the yeah, this is great achievement and exciting research. And I think we will have a lot of questions, I hope. So who will be starting? Is there any questions?
Thank you. While folks are thinking over the amazing talk you just gave, I thought I might start out with a couple of questions. So is the kinase not a GARD-E, or can it be a GARD-E, thought of as a GARD-E, based on your earlier talk?
Yes. So what sort of model does it fall into? It's a good question. I would say so. We're treating it more as an integrated decoy. So we suppose that the effector is targeting something else in the genome. It might be another kinase, and this SR62 kinase has become a trap, and is able to trap the effector into triggering an immune response. But this is not something we have proven. This is a hypothesis, and I think we would have to understand what the effector is interacting with in the proteome, in addition to the kinase fusion protein.
Right. So I wanted to ask you, following that, like, is the kinase activity important for how it works, or?
That's a very good question, and it was something the reviewers asked for also, and we blushed over that and said that this was beyond the scope of the paper. But we are now studying that, and we have found that kinase 1 has strong kinase activity in vitro, and kinase 2, on the other hand, does not have kinase activity in our assay, and this is what you would expect because it's missing some of the important residues in the kinase. We're beginning to unravel the role of this kinase activity. We found that it phosphorylates a particular residue in kinase 2. And if we knock out, if we replace that residue with an amino acid, an alanine, I think it is, then we abolish function. We don't yet understand how this is important for activating the NLR.
Thank you.
Yeah, thank you again for your second nice talk. So decoy, if I have understood correctly, must acquire specificity to a certain effector. And of course, you did not go so much into detail, but probably you, or very likely, you have looked at the kind of generic structure, how this specificity can be attained, like leucine-rich repeats or other kind of antibody principles where modules are shuffled to create a whole variety of possible specificity. And
is this the great variability that we see in the extended beta finger? Is that under diversifying selection to create a scaffold? Is it a shapeshifter that can interact with effectors? That is a direction of travel that we are exploring.
Go ahead.
You put your finger right on it, Jochen.
Yana savollar bormi? Agar savollar bo'lmasa, bu yerda bir narsani aytib o'tmoqchi edim. Agar prezentatsiyada e'tibor bergan bo'lsanglar, juda ham ko'p o'sha nature maqolalarni ko'rsatdi. Bularda o'rganadigan narsamiz ularni o'sha qilgan ishiga biz hali yetishimizga ancha bor. Lekin bularda yana boshqa narsa bor. Shunchalik zo'r ishlar qilib qo'ysa ham bulardagi oddiylik, biz nima desam, bizadagi o'sha qanaqadir odamgarligi, o'sha nima desam, siz qanaqa yordam so'rasangiz bular jon dildan sizga yordam berishligi, oddiyligi, odamiyligini saqlab qolgan holatda o'sha nima desam, bu narsa juda ajoyib, biz o'rganadigan narsa deb o'ylayman. Chunki bizada agar shu darajada maqola qilsa, bizada o'zidan katta qanaqadir ma'lumot nimadir yasab oladi. Men hozir buni ingliz tilida aytmayotganim, olimlar ichida Nature'da chiqarmaganlari bor. Hozir maddohlik bo'lib ketmasin deb bu narsani faqat o'zbek tilida o'zim gapirdim. Thank you.
Thank you.